Now, with this insight, we can individualize the care for each patient. So examples of technological tools or domains that are ushering us into the era of precision medicine. For instance,
is before you get that, but it is not only a single mutation that will lead to cancer. We have something we call an initiator mutation. So the mutation, the first mutation that will cause the cell to get transformed.
Look back in our own setting, and we're only limited by just the traditional screening methods, which is the self-examination or the mammogram and ultrasound scan, and for those who can afford the MRIs. And that is sometimes even when it's almost too late to do anything about it.
These are also currently being used to detect, how your transcriptome levels are contributing to you being predisposed to cancer.
So, for instance, there's a chemical which we want to detect. Okay, there's a chemical we want to detect. We detect that chemical by the chemical binding to a receptor. Okay, so this chemical will actually, the right name is an analyte. So the analyte will bind to a receptor. The correct name is a bioreceptor. Okay, when the analyte binds to the bioreceptor,
the less frequent use of unnecessary tests in our patients. So then there's no more overdiagnosis and overtreatment, because some patients definitely do not have it, but you're overdiagnosing them.
Present with a very poor clinical pathological features such as larger tumor size, advanced tumor grades. They present with high Nottingham prognostic index, high clinical clinical prognostic stages.
Well, lots of points of care have been used as primary screening, but there is none to accurately tell you that this particular marker means that 100%, 95% you have this cancer.
That this particular disease that the patient has, is it aggressive based on how the vascular system of that tumor is? You'll be able to predict. You can also look at metabolic activities there. Is there high uptake of metabolites? And then you just manage your patient.
It leads to the production of a signal. Okay, it leads to the production of a signal. The signal that is produced from the analyte binding to the bioreceptor can be changed and can be essentially measured. Okay.
And then we need a series of mutations that is called the driver mutations. And these driver mutations are a number.